good evening. Welcome to Aakash BYJU'S NEET channel. I'm your teacher Ankita and I welcome you all in today's class where we will be discussing about the very important organelles of the cell. We will be tackling the nucleus, the brain of the cell. We'll be discussing about the cilia and the flagella and of course the other important parts. So I hope that all of you are ready for the class. I hope that everyone, all of you are excited for this interesting class. This is the last class for the chapter Cell, the Unit of Life. So with this particular session, we will be actually finishing our first chapter, right? And it's such an interesting thing because this chapter is really very important and that the chapter is not very difficult, I would say. So we have accomplished something today. Good evening, everyone. Good evening, Vaishnavi. Good evening, Mirender, Lavanya, Deep, Manisha. Good evening, everyone. How are you all? I hope that all of you are doing good and in a great health and are ready for the session. Very good. I hope that everyone, I'm clearly audible and visible to each one of you. If that's so, please make sure you hit the like button for the video and do write in the comment section below also. And if you're new here, do take a moment and subscribe to our channel where we make sure to give you the best quality education that will be helping you to ace your examination. Yes, everyone, good evening. I'm good. How are you all? So let's move ahead, right? Now, this is the last part. This is the part 6 of the chapter, the cell, the unit of life. Now, we have these all classes. Everyone, do take a look over here, right? If you have missed any of these classes, you can go back to our channel, right? You can look for the playlist, and of course, in the playlist, we have all of these videos. What we have like total of 5 parts. A clear-cut differentiation, what we have over there. So, everyone, are you clear with this? Yes. How's the Josh, everyone? The week is almost, the week will just go as of now, right? It's Thursday today and of course we'll have Friday and yeah, then Saturday, Sunday. But everyone, those are the days where we have to study more. Yes? Good evening, everyone. Good evening. I can see some hearts popping up. Thank you so much, everyone. Hello, Aditya. Hi. Yes. Okay. Is it noisy? Okay. I will ask the team to just see. Yes, I'll ask the team to see through it. Okay. Is it better now? Everyone, my voice is clear. Is it better now? I hope that there's no disturbance from our side. Very good, very good. Chaliye. So let's start our class, everyone. We will be first, of course, we'll be discussing about it. Today we have a lot to study, right? We'll be discussing about the cytoskeleton and of course we will be discussing about the cilia and the flagella. We have centrosome and centriole, right? And of course we'll be finishing our class with the nucleus and microbody. So we have a lot on our plate today. So everyone, let's get started. So we have some questions. Yesterday I didn't have, the questions for us to revise. So here everyone. Let's take a look at the question and quickly answer over here. The inner membrane of the mitochondria bears folding finger-like projections. These are what? These actually help in the increasing the, increase surface area, increase thickness of mitochondria, keep all the substance away, or increase protein oxidation. Very good. Question number 1 is, of course, the one answer is, it actually helps in increasing the surface area, right? So we have discussed that in mitochondria, we have the inner folds, right? And these inner folds actually help in increasing the surface area for the production of ATP. Very good. Next question, everyone. Which of the following plastids store fat? Okay. Which of these plastids store fat? Jaldi se everyone. So you remember we discussed about the chromoplast. In leucoplast we have discussed about. Yes. Very good, very good. I can see 2B answer yes. Very good. A myeloplast will be playing a very important role in the storage of the carbohydrates. Very good, everyone. Chromoplasts, of course, have the color pigment and leucoplasts in general plays a very important role in the storage. Chaliye. Next question, everyone. Mitochondria. Mitochondrial DNA is what? Is it naked? Is it circular? Is it double-stranded? It's all of the above. Interesting question, right? This is a clear-cut question from our NCERT. Everyone, please open your NCERT. Mitochondrial DNA is what? It's there in our NCERT. I can see some wrong answers. Right, I can see some wrong answers. Think about it, everyone. Seems like this, I'll not be giving you the answer for this question. Seems like this, you have to look at the NCERT. So everyone, it's a homework question. I will leave you with this homework question, right? Putting it on a star, and I want all of you to find the answer for this question. Right? It's a direct line from our NCERT. So take a moment and find the answer. Okay? Chaliye. I will move ahead to the next question. Ground substance present inside the mitochondria is called as what? What we call it as? Stroma, matrix, cell sap, or cytoplasm. See, this seems like this is a very easy, very good. It is called as matrix. Very good, everyone. So we had these interesting questions with us, right? And now we have seen all of these. We'll be moving to the theory part of our class today, everyone. Now I want all of you to pay attention to the class, right? And everyone get really focused. Yes. Okay, chaliye. Yes, I will wait for the question number 3 ka answer, right? Just go and search for it and come back and tell me, or you can write in the comment section below later. Chaliye, good to go, right? So everyone, let's get started with it. So of course, what we have? Okay, Rajneesh. Right? Now we all know that we have in our body, we have the skeleton, right? We also call it as it plays a very important role and it acts as a structural framework, skeleton framework. We use the word framework because of this skeletal only, right? We have the structure, right? We have muscles attached to it, and of course we can move our body, and this is how we will look. Now if we don't have this, yeah, we are nothing but a mass, right? Some muscles here and there. So in cells also we have something called as cytoskeleton, which plays a very important role in the framework, right? It actually provides a framework to the cell. And of course, what are they? They are the filamentous structure, thread-like structure. धागेदार स्ट्रक्चर है ये, ठीक है? चलिए आगे बढ़ते हैं इनके बारे में. तो व्हाट इज साइटोस्केलेटन? एवरीवन, क्या है स्केलेटन? साइटोस्केलेटन क्या है? दे आर दी फिलामेंटस स्ट्रक्चर, थ्रेड लाइक स्ट्रक्चर्स हैं ऐसे, है ना? प्रोटीन्स के होते हैं, कंसिस्ट ऑफ 3 वेरी इंपॉर्टेंट स्ट्रक्चर दैट वी विल बी डिस्कसिंग मोर फर्दर. माइक्रोफिलामेंट, इंटरमीडिएट फिलामेंट एंड माइक्रोट्यूब्यूल्स. हम एनसीईआरटी रीड भी करेंगे तो चिंता की कोई बात नहीं है. अगर हम यहाँ पे स्ट्रक्चर देखें, राइट, सो वी हैव द साइटोस्केलेटन. कैसे दिखते हैं ये? माइक्रोफिलामेंट यहाँ पे दिख रहे हैं आपको, थ्रेड लाइक स्ट्रक्चर्स हैं, फिलामेंटस स्ट्रक्चर्स हैं ऑल टुगेदर अगर हम बोले. फिर वी हैव इंटरमीडिएट फिलामेंट एंड ऑफ कोर्स वी हैव द माइक्रोट्यूब्यूल्स. इन तीनों के बारे में हम डिटेल में पढ़ेंगे. स्टार्टिंग कर रहे हैं हम इंटरमीडिएट फिलामेंट से. अब इंटरमीडिएट फिलामेंट, इंटर वर्ड आ रहा है बीच में, राइट? इंटरमीडिएट. सो ऑफ कोर्स इसका कोई रीजन होगा, हम जानेंगे इसका रीजन क्या है. एवरीवन, आई होप दैट ऑल ऑफ यू आर देयर विद मी, right? And I will be teaching you in both in English and in Hindi. We'll try to have a balance of it, so don't worry. Clear, everyone? So what we have? We have the intermediate filament and they have a diameter of 10 micrometer. Very, very important. All the numbers that we have are super important over here, right? Now, the diameter of this intermediate filament is kind of in between the microfilament and the microtubules. That is the reason they're basically in between. They plays a very important role and provides a mechanical strength to the cells and to the tissue. They are hollow, right? That's a very important thing you can note down. They are hollow and of course they are made up of, like we can say that they are the non-contractile protein. Contractile protein क्या होता है? Anyone can you tell me what is contractile protein? Contract हो रहा है, relax हो रहा है. So of course, with the word non over here, we can say that it will not be able to contract. Yes or no? Yes? Good. Right? This plays a very important role. Of course, we know that it plays a very important role in the mechanical strength. अब दूसरा हम बात करेंगे जो बहुत स्पेशल है, जो कि क्या है, माइक्रोफिलामेंट. माइक्रो वर्ड, माइक्रो छोटा होता है, राइट, माइक्रोफिलामेंट. सो वी कैन क्लियरली सी दैट द माइक्रोफिलामेंट का डायमीटर इज लेस एज कंपेयर टू द इंटरमीडिएट फिलामेंट. इफ यू लुक ओवर हियर, द डायमीटर ऑफ द माइक्रोफिलामेंट इज 7 नैनोमीटर. वेरी वेरी इंपॉर्टेंट. दे आर मेड अप ऑफ एक्टिन प्रोटीन, मायोसिन प्रोटीन, राइट, जो मसल्स में है हमारे. सो नोट दिस डाउन एवरीवन. Actin and myosin protein, and they plays a very important role in the cell movement and the cell division. Clear? ये बहुत छोटी सी इंफॉर्मेशन है, but इस पे क्वेश्चन आ सकते हैं. So we will remember as it is. दूसरा, very important, over here we can see the diameter, right? And last, but not the least, we have the microtubules. Now look at the diameter of them, right? They have a largest diameter among 3 of these slot. The smallest one is microfilament, then we have the intermediate filament, and then we have the microtubules. Now of course they are hollow, right? थोड़े से hollow हैं. Clear? Microfilament was solid. ये hollow हैं, and of course they have a cylindrical kind of shape, ऐसा है, right? And of course they have a very special protein, tubulin. This is very very important, everyone, right? This is very very important. This microtubules will be studying in upcoming topic, centriole. Centrosome का जो structure है हम उसमें पढ़ेंगे सबके बारे में, ठीक है? तो ये बहुत जरूरी है for all of us. ये we have which protein? We have the tubulin protein. Provides both mechanical support and the cell movement. Plays a very important role in the mechanical support and in the cell movement. And we can add help in the formation, right, of the centrosome. हम आगे देखेंगे. They plays a very important role. Clear? So ये तो हमारे पास में हो गया brief introduction, I would say, about the cytoskeleton and its different types. Microfilament, microtubules, so we have 2 chains, right? We have alpha and beta. Clear over here? That is written over here. And with this, let's take a look over here about that. They can assemble. They can become big, and of course they can deassemble also. So they have a very interesting property, right? Because what they are the contractile protein. They can contract and relax. So this is a very important features of the microtubule. Now that we are done with this, everyone, we can quickly summarize the function of the cytoskeleton. What is the main function of the cytoskeleton? Provide the mechanical support, helps in the motility, right? Helps in the organelles to move within the cell. So the organelles that are in the cell, right, they can actually move with the help of the cytoskeleton. Maintain the shape of the cell and helps the cell division. So if we have to take a look at the chotu, so everyone look at this topic, right? In our textbook, we just have for 4 or 5 lines. Network of the filamentous proteins structure consisting of microtubules, microfilament and intermediate filament present in the cytoplasm, collectively called as the cytoskeleton, plays a very important role in many functions such as mechanical support, motility and maintenance of the cell, maintenance of the shape of the cell. Clear everyone? जल्दी से बता दीजिए. All of you, this was the easiest topic that we have. सबको समझ में आ गया? साइटोस्केलेटन बस इतनी लाइन लिखी है जितना हमने पढ़ लिया, हम उसे और डिटेल में पढ़ेंगे. बट एवरीवन, आई होप दैट दिस इज क्लियर टू ईच वन ऑफ अस. बच्चों कैसे हो? राइट, आई कैन सी. गुड गुड बच्चों, प्लीज कीप द जोश हाई, हैव द एनर्जी इन क्लास एंड वी विल बी एबल टू फिनिश दिस ऑफ रियली वेरी क्विकली. चलिए, राइट, ओके. नाउ लेट मी आस्क यू, देयर आर फ्यू मोर स्केलेटन्स दैट वी हैव, राइट, जो सेल में है, जो एक्चुअली, राइट, वो किसके बने हुए हैं? दे आर मेड ऑफ द साइटोस्केलेटन एलिमेंट. नाउ व्हाट विल बी लुकिंग एट एवरीवन? विल बी लुकिंग एट के व्हाट आर द डिफरेंट स्ट्रक्चर्स जो हमारे साइटोस्केलेटन एलिमेंट्स के बने हुए हैं. क्लियर? वेरी गुड संचिता, मिलरेंद्र, तनु, कुणाल, वेलकम टू द क्लास. ओके, चलिए. नाउ लेट्स टॉक अबाउट द 2 वेरी इंपॉर्टेंट स्ट्रक्चर एवरीवन, सेंट्रोसोम एंड सेंट्रियोल. हम ये पहले पढ़ेंगे, उसके बाद में वी हैव द सीलिया एंड द फ्लैजेला. राइट, चलिए. ओके, व्हाट इज सेंट्रोसोम? लेट्स गो बैक. लेट मी आस्क यू, व्हाट इज सेंट्रोसोम एवरीवन? जल्दी से जस्ट राइट वन सिंगल लाइन. क्या होता है? आई एम श्योर आपको पता है क्या होता है सेंट्रोसोम और क्या है सेंट्रियोल. हम बोलते हैं सेंट्रियोल इज सुपर इंपॉर्टेंट, राइट. सेंट्रियोल हेल्प्स इन सेल डिविजन, है ना. तो सेंट्रियोल और सेंट्रोसोम का क्या कनेक्शन है? ओके, सेंट्रियोल सेल ऑर्गेनेल हो गया. ओके, वेरी गुड. और क्या है? सेल डिवीजन में हेल्प करता है. वेरी गुड. और व्हाट वी हैव अदर इंफॉर्मेशन अबाउट दिस? और क्या इंफॉर्मेशन है? यस, ओके, वेरी गुड. Centrosome is the nucleus of the centriole. Center part. Two centrioles arranged in 90 degrees formed from the cytoskeleton. Very good. Very good, everyone. So we have some basic information about it, right? ये क्या है, कैसे काम कर रहा है? Let's see more further. So हमें क्या पता है? Centrosomes हमें हमेशा सिर्फ animal cells में मिलते हैं, plant cells में नहीं है. So we can write that they are only found in the plant cells and are absent in the plant cells. ठीक है? They are usually found very close to the nucleus. See, we can see over here. They found very close to the nucleus and the centriole are kind of cylindrical structure. है ना? Clear? They are kind of in a cylindrical structure. Yes, everyone? Clear? अब यहाँ पे जब cylindrical structure बोल रहे हैं और ये बोल रहे हैं कि दो perpendicular ऐसे हैं, they are made from the two units. Right? This is how it is arranged, everyone. Clear? This is how it is arranged. और इसे हम क्या बोलते हैं? This is perpendicular. Right? This is perpendicular and what we call it as, we have a very interesting name for it. We call it as the diplosome. Yes. Clear, everyone? You can note down this name. What we call it as? We call it as the diplosome. Clear, everyone? Very good. They are perpendicular, right? They are perpendicular. चलिए, this is a very important information. Please take a note, and this is how they will look. See over here. ये तो हमने थोड़ा सा ऐसे यहाँ पे कर दिया. Clear? Very good. See over here. Centrosome. दो हमारे पास में centriole हैं. When they come together, they form the centrosome, और जिसे हम diplosome भी बोलते हैं. Clear? Here we have. They lie perpendicular to one another in this structure that we have. If you look over here, everyone, they are perpendicular at right angle. ठीक है? ये हमें याद रहेगा. Then, if you look over here, अगर हम इस structure को ध्यान से देखें, right, जो arrangement है कैसा है इनका? It's in a shape of cartwheel, surrounded by the amorphous pericentriole protein. ठीक है? अब ये cartwheel arrangement है, हमें ऐसा दिखता है, और इसके around we have something which is not very definite in shape, right? Clear? इसमें कोई clear cut division नहीं है, कोई defined shape नहीं है इसकी. Clear? ये इसको हम लोग ये बोल रहे हैं, right? Pericentriole protein. Clear? Peri, right? क्योंकि ये बहुत, इसमें क्या है? It is a matrix and of course it's a matrix of protein. Clear? And इसमें क्या है? हमारे जो centrioles है वो embedded हैं. Very good, very good. Clear? So कौन सा मटेरियल है? ये मटेरियल है, ये प्रोटीन है हम बोलेंगे. And they consist for the proteins. इनके पास में प्रोटीन्स है जो हेल्प करेंगे ट्रांसपोर्टेशन में. अब हम इसका स्ट्रक्चर ध्यान से देखेंगे. I can see that all of you are well prepared for the class. सब अच्छे से पढ़ के आए हैं, सब लोग आंसर कर रहे हैं, तो I am very happy. Let's talk about the centriole. Centrosome के बारे में पता चल गया. दो centriole का बनता है, perpendicular है इस तरीके से, right? एंड ऑफ कोर्स हमने क्या पढ़ा उसके बारे में कि इसको दूसरा नाम डिप्लोसोम है एंड परपेंडिकुलर है, राइट? ये हम बात याद रखेंगे. अब हम पढ़ेंगे सेंट्रियोल के बारे में. विल बी लुकिंग एट द स्ट्रक्चर ऑफ इट कि हाउ इट इज फॉर्म्ड. द सेंट्रियोल ओवर हियर, कैन वी सी ओवर हियर, राइट? द सेंट्रियोल दैट वी हैज 9 पेरिफेरल फिब्रीन. बेसिकली यहाँ पे क्या है? ट्यूब्यूल्स. ट्यूबुलिन याद है आपको? ऑल ऑफ यू, आई एम श्योर यू रिमेंबर दैट, राइट? अभी पढ़ा. सो ये जो हमें दिख रहे हैं ना, these triplets, let me just change the pen color over here. The triplets that we see over here, right? 1, 2, 3, all of these, right? Clear, everyone? ये 1, 2, 3, 4, 5, 6, 7, 8, 9. These 9 small circles that we have over here, right? They are actually made up of the tubulin protein, which is a microtubule. Everyone, are we clear? Yes, are we clear with this? किसके बने हैं ये? ट्यूब्यूलिन के, जो अभी हमने पढ़ा था किसमें? माइक्रोट्यूब्यूल में. वेरी गुड. नाउ दैट वी हैव दिस अरेंजमेंट, राइट, व्हाट वी हैव इन दिस? A, B and C, right? We name them as A, B and C. हमें क्या पता है? ये फिब्रिल्स किसके बने हैं? ट्यूब्यूलिन के. और देर अरेंज इन ट्रिपलेट्स, 3 हैं. 1, 2, 3. We can see right over here A, B and C. These A, B and C are kind of linked. So C is linked with the A. Clear? A, B, C, A, B, C. So C and A are linked. इसे हम AC linker भी बोलते हैं. Clear everyone? What we call it as? We call it as a AC linkers also. Clear? AC linkers. Clear over here? जो actually join कर रहे हैं C and A को. Very good. AC linkers, absolutely correct. चलिए, now that we are clear with this, let's talk about more. See, यहाँ पे आप देख लीजिए everyone, right? You can take a screenshot of it also. We have tubulin triplets हैं हमारे, connecting fibril है यहाँ पे. We can see they are connected also. And हमारे पास में linkers हैं. Now, अब हम बात करेंगे center की. In the center what we have is the hub. जो center में है उसे हम क्या बोलते हैं? hub बोलते हैं. और ये जो है, जो attach है ट्यूब्यूलिन से सेंटर में, हब में अटैच है. कैन यू नेम देम? व्हाट वी कॉल देम एज? यस, वेरी गुड. सो वी कैन सी दीज structures, right? They are attached to the hub and they're actually connecting these triplets, microtubules, to the hub. What we call them as? Can I give you hints starting with S? Very good, Kunal, very good. They are called as the radical spool. Clear? Yes. Okay, चलिए. So this is a very important thing again. So we can see that they are connected, right? They are connected. Hub जो है वो इन tubules के साथ में कैसे connect है? With this structure which is the radial spokes. Done? Easy है. Recall करना पड़ेगा बार बार. I know that it's a little bit difficult, but इसे याद करेंगे तो हो जाएगा. And of course there are no tubules in the center. Center में कोई भी हमारा ट्यूब्यूल नहीं है. राइट? अब यहां पे फटाफट से देख लेते हैं. अरेंजमेंट इज व्हाट? 9 plus 0. 9 plus 0 means in the center they don't have anything and at the side what we have total of 9. पूरे चारों तरफ क्या है हमारे पास में? 9 है. 9 ट्यूब्यूल्स हैं. सेंटर में कोई भी ट्यूब्यूल नहीं है. अब हमारे पास में 2 सेंट्रियोल हैं. Right? Now we have 2 centriole. So can you tell me what will be the total number of the tubules? जल्दी से बताइए एवरीवन. 3 into 9 करेंगे तो what will be the number? कुनाल, you're asking मैम centriole lower plants like algae में ही पाए जाते हैं क्या? Think about it. We know that centrioles are only present in animals. Animal cell में, right? Yes or no? So I think you got the answer. Everyone quickly बताइए, अगर 3 हमारे पास में ट्रिपलेट्स हैं और टोटल कितना है उनका? 9 इन नंबर हैं, राइट? तो क्या होगा? व्हाट इज द टोटल? अगर कोई आपसे पूछेगा एग्जामिनेशन में, इफ दिस क्वेश्चन कम्स दैट व्हाट इज द टोटल नंबर ऑफ द ट्यूब्यूल्स दैट आर प्रेजेंट इन द सेंट्रियोल, व्हाट यू विल से? 27. दैट इज 1, राइट? एक सेंट्रियोल के अंदर हमारे पास में कितने हैं? 27. व्हाट अबाउट द अदर सेंट्रियोल? इट विल हैव द सेम थिंग. 27 plus 24, 27 plus 24, 27 कितना होगा? Yes? 54. So the total number of the tubules that are present in a centrosome, because it has 2 centriole, the number is 54 का arrangement, because center में, hub में we don't have any tubules. Very good. चलिए. This is the hub, this is the spokes. Okay. Now what is the main function of it? We all know that the main function of the centriole is during the cell division. They help in the formation of the spindle fiber. especially in the animals because they're there in the animal cells and they plays a very important role in the cell division. तो centrosomes, right? किसका बना है? दो centriole का. Centriole क्या function है? To produce the spindle fibers during the cell division, right? Clear everyone? In plant cells we know that centriole are absent, but they appear, right, to nucleate near the nuclear membrane, right? We'll remember this. All of you, are we clear? Yes. Quick thumbs up. Very good. More functions. The second function, it's from the basal body of the cilia and the flagella, which we'll be discussing in an upcoming topic, that they plays a very important role in the formation of the basal body, right? जो part, जो cell के, जो basically एक organism के साथ attach है, this part is formed by the centriole. ठीक है? चलिए, everyone, are we clear with this? Quick thumbs up. Yes, and everyone, how many likes we have? Please make sure you hit the like button for the video, right? Good. Everyone quickly hit the like button. Yes. So we are done with the functions of the centriole now. Now we'll be discussing about the cilia and the flagella. Now we know that cilia and the flagella are a very important, which organ? Basically they play which important part? Cilia and flagella have a very important role in the movement, right? In the locomotion. They plays a very important role in moving the organism from here to here. Yes? So now we'll be discussing about the eukaryotes. And are they really stationary? We know that they are not, right? We know that the eukaryotic cells are not all the eukaryotic cells are stationary. We can see the movement happening. See over here the cilia? And over here we can see the function, right? We can see the flagella. So these two plays a very important role in the movement or the locomotion of the cell. So we have two types of it. We have cilia and we have flagella. Of course we have the cilia over here and flagella in the euglena, and we have the paramecium over here. Clear? अब हम cilia की बात करेंगे, and this is something which I really very interested in. Look at these people, right? I'm sure आपने कहीं ना कहीं मूवीज में या फिर कहीं ना कहीं डॉक्यूमेंट्रीज में देखा होगा. This is how the people are actually pushing the boat. I'm sure अगर आप केरला से हैं तो you would have seen this. And in movies of all, if you're not from the Kerala also, you would have seen this, right? क्या हो रहा है? नाव में लोग बैठे हुए हैं. They have the, जैसे हम हिंदी में चप्पू बोलते हैं, right? और they are using it to push. पानी को पीछे धकेल रहे हैं और बोट आगे जा रही. Similarly, the cilia work in this particular way. The way they are moving ahead by pushing the water backward, this is how the cilia works. Everyone, are we clear? Right? So, they are, if we talk about the cilia, right, they are short. They are not huge. They are very small structures, very, very fine structure that they are. Here, like, they'll just look like a hair, right? And, or like movement. So, they just push the water backward and move ahead. Yes. Do they use a third law of motion? Think about it. Equal, every action has an equal opposite action. मतलब जितना action है उतना reaction हो रहा है. Yes. Think about it. Medium भी move हो रहा है, and of course the cell is also moving ahead. Clear? Yes, very good. So this will remember about the cilia. They're short. They have the hair-like structures, right? They look like a hair-like structures, and of course they move. They plays a very important role in the movement. Now, if we quickly call the flagella, yes, flagella, we know that it's an extension of the cell membrane. Cell membrane पे रहता है. If you see, everyone observe this GIF. See, this is the structure, right? It is actually embedded in the plasma membrane or the cell membrane. So it is a motile. It is present in the motile bacteria. It's definitely longer than the cilia, right? And it is made up of the protein which is called as the flagenin. Important thing, everyone. Please do remember this. And this is the longest portion of the cell body. So for example, if it is present in the bacteria, right? This flagella will be the longest portion of the cell body. Clear? Yes. Chandan Yadav will discuss that. आप बताएं क्या कोई फंक्शन नहीं है उसका तो वो तो डेंजरस है, राइट? ओके, चलिए Now let's move ahead and let's quickly recall more about the flagella. They are longer, they are whip-like structure, but यहाँ पे जो movement हो रहा है वो सिर्फ movement है, right? It is not pushing any medium. जिस तरीके से सीलिया आगे चीजों को पीछे धकेल के आगे जा रहे थे, flagella are not doing that. They're just moving straight forward. So in prokaryotic flagella, right? We know that that's an action. But when we talk about the prokaryotic flagella and the eukaryotic flagella, they are different. This is an important point for all of us to remember. Okay, they're different from each other. Clear, everyone? I hope that all of you, are you clear with this? Right, they're different from each other. Now, if you look over here, right, this is the flagella, and if you look closer, see this particular part. Can you call it what we call it as? कहाँ पे attach है? We have studied this in the prokaryotic cell का structure में. There are 3 parts of it, right? There are 3 parts of the flagella. Can anyone tell me the filamentous part was there, head part was there, and what was the other part? Hook. Yes, जल्दी से बताइए, all of you. What was the 3 parts of the flagella? Basal body, okay. Yes, जल्दी से everyone. Okay, basal body I can see, बढ़िया है. What else? Jaldi, jaldi. We have hook and we have the filament, right? So over here we can see that it is the extension of the plasma membrane, right? And it actually arises from the basal body. Basal body say it arises. And now we know that the basal body कैसे होते हैं? They are cylindrical. So I'll write over here. They are cylindrical. Okay, and they are the one that contains 9 triplets. अभी हमने पढ़ा है ना? Right, they are cylindrical. हमने अभी पढ़ा था कि the centrioles. Yes, the centrioles. If you go back and see, the centrioles help in the formation of the basal body of cilia and flagella. So this is very, very important. Clear? So basal body है जहाँ से हमारा flagella बन रहा है, और basal body कहाँ से बन रही है? Centriole. Basically it forms. Centriole forms a basal body. So here we have an important information for all of us. Clear? चलिए. With this everyone, we have a very interesting next topic now. Yes. Kunal, I'll answer your question. I'll answer your question. Give me some time. I'll answer your question. अब are we clear up to here everyone? अब हम क्या देखेंगे? अब हम structure देखेंगे इसका. Right, we will be looking at the structure of the cilia and the flagella, the basal body का structure basically. अब हम देखेंगे कि अंदर का क्या structure है. Now we know that कि basal body, flagella is arising from the basal body, and centriole plays a very important role in the formation of the basal body. So we can see there is some connection over here. जो हमने 3 triplets वाला structure पढ़ा, right? It has some similarities, but not exact the copy. So let's see what is the structure over here. Now, if you look over here, we have a very strong core which is called as the anoxome. Clear, everyone? All of you. थोड़ा सा difficult नए words आ रहे हैं. I know that for all of you there are few new words that has been added into your vocab, but आराम से अगर आप बार-बार पढ़ोगे तो याद हो जाएगा. Clear? इस पूरे एरिया को, the core area, right? Their core. ये जो आउटर बाउंड्री है और इसके अंदर का ये सब है, इसे हम क्या बोलते हैं? एनॉक्सिमी. क्लियर एवरीवन? क्या बोल रहे हैं हम इसे? 1 मिनट. हाँ, क्लियर क्लियर. क्विक थम्स अप जल्दी से. वेरी गुड. राइट, नाउ दिस हैज अ वेरी सिमिलर स्ट्रक्चर टू दी जो हमने कार्टव्हील स्ट्रक्चर पढ़ा उससे ज्यादा. अब हम देखते हैं क्या सिमिलैरिटीज हैं. सेंटर में ऑफ कोर्स, नाउ द मेजर डिफरेंस दैट वी हैव फ्रॉम देयर टू हियर कि सेंटर में वहां पे हमारे पास में कुछ नहीं था, राइट? इन द हब, राइट, इन द सेंट्रल हब वी डिडंट हैव एनी माइक्रोट्यूब्यूल्स, बट हियर वी हैव अ पेयर ऑफ ट्यूब्यूल्स. ठीक है, व्हाट वी हैव अ पेयर ऑफ ट्यूब्यूल्स लाइक दिस. Central pair of microtubules are present. Then of course, instead of the triplet, we have the doublets. Can you see over here? पहले वहां पे A, B, C था, अब यहाँ पे सिर्फ A and B है. Clear? अब वहां पे हमारे पास में क्या है? सिर्फ A and B. Again, total number क्या है? 9. We can clearly see. We have 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 doublets हैं of microtubules, circularly arranged है. और यहाँ पे हमारे पास में मेम्ब्रेन है। पहले वाले में मेम्ब्रेन नहीं थी, पर यहाँ पे वी हैव दी प्लाज्मा मेम्ब्रेन। रिमेंबर दिस एवरीवन, क्लियर? यस, याद रहेगा। 9 डबलेट्स है हमारे पास में माइक्रोट्यूब्यूल्स के, सेंटर में 2 है तो 9+2 अरेंजमेंट। 9+0 कहां पे है? सेंट्रियोल में। और 9+2 अरेंजमेंट कहां पे है? सीलिया एंड फ्लैजेला में। याद रहेगा ना हमें? यस, बहुत इजी स्ट्रक्चर है अगर हमें समझ में आ जाए तो, है ना? एंड आई होप दैट आप सबको समझ में आ रहा है। आई होप दैट ऑल ऑफ यू आर थ्रू विद इट। चलिए, नाउ दैट वी आर क्लियर विद दिस, लेट्स सी द 9+2 अरेंजमेंट। प्लाज्मा मेम्ब्रेन है, जैसे मैंने अभी आपको बताया। सेंट्रियोल में प्लाज्मा मेम्ब्रेन नहीं थी, ओपन स्पेस है, राइट? कोई भी शेप नहीं है बेसिकली, नो देयर इज नो डेफिनेट शेप। यहाँ पे वी हैव द प्लाज्मा मेम्ब्रेन, इनर शीट भी है, यहाँ पे रेडिकल स्पूक हैं, स्पूक्स हैं, वो तो रहेंगे ही। 9+2 अरेंजमेंट है हमारे पास में। सेंटर में वी हैव द माइक्रोट्यूब्यूल्स, इसलिए 9+2 अरेंजमेंट है। वी हैव द लिंकर्स ऑल्सो, राइट? दीज आर द हेड्स, राइट? डायनेन हेड्स हैं यहाँ पे, सी ओवर हियर, क्लियर? बस इतना याद रखेंगे। ये आर प्रोटीन्स, राइट, which used for the ATP to drive the movement of the cilia and flagella. ठीक है everyone? Linkers join कर रहे हैं, ये सब हमारे पास में है। I'll write this function over here, right? So they are protein which uses ATP. ये क्या? ये वाले प्रोटीन ATP को यूज कर रहे हैं to drive, everyone, I hope that you are there with me, right? To drive the movement of cilia and flagella. Okay? You can take a screenshot of this. Everyone, are we clear? Yes, are we clear with this? So with this what happened? What this? क्या हुआ हमारा? We have looked at the structure of the centrosome, centriole, cilia and the flagella. एग्जाम पॉइंट ऑफ व्यू से इंपॉर्टेंट है, PYQs आते हैं तो हमें याद रहेगा। क्लियर? ऑल ऑफ यू आर वी क्लियर विद इट? थम्स अप। चलिए, लेट मी आस्क यू क्वेश्चन एवरीवन। कैन यू नेम अ सेल इन ह्यूमंस विद अ फ्लैजेला? क्या हमारे ह्यूमंस में, बी द मेल और द फीमेल एंड एनी ऑफ द जेंडर, डू वी हैव अ सेल व्हिच हैज फ्लैजेला? वेरी गुड अक्षत, बहुत जल्दी आंसर आ गया। Very good, very good. Mehbooba, yes. Lofi, very good. Yes, the correct answer is the sperms. So sperms have the flagella. Chaliye. One more question time everyone here. Which among the following is found only in the animal cell? Just write the option in the chat box everyone. Which among the following only found in the animal cell? सेंट्रीजोम, सेंट्रियोल, प्लाज्मा मेम्ब्रेन, साइटोप्लाज्म, राइबोसोम्स। क्या इनमें से सिर्फ एनिमल सेल में मिलता है हमको? सेंट्रोसोम एंड सेंट्रियोल। वेरी गुड, ऑप्शन नंबर ए इज द करेक्ट आंसर। नेक्स्ट क्वेश्चन एवरीवन, सीलिया एंड फ्लैजेला शो व्हिच काइंड ऑफ अरेंजमेंट ऑफ माइक्रोट्यूब्यूल्स? सीलिया एंड फ्लैजेला में स्पेसिफिकली दे आर आस्किंग व्हिच काइंड ऑफ अरेंजमेंट ऑफ माइक्रोट्यूब्यूल्स वी कैन सी। हमने 2 पढ़े हैं, 9+0 और 9+2। ये 2 तो ऑप्शन में होने ही नहीं चाहिए, राइट? एंड द करेक्ट आंसर इज 9+2। 9+0 हमारा सेंट्रियोल में है। चलिए, so with this everyone, let's quickly revise what we have studied. We have studied about the cytoskeleton organelles, right? Centrosome, centriole, cilia and the flagella. Now everyone, अब हम एक बार कैन वी क्विकली गो थ्रू दिस टेक्स्ट बुक का, एनसीईआरटी का लाइंस, राइट? फटाफट से पढ़ते हैं। वी हैव सम टाइम, वी हैव टू फिनिश द न्यूक्लियस। इसके बाद में न्यूक्लियस इज द टॉपिक विच विल टेक सम टाइम, तो फटाफट से इसको पढ़े। सीलिया, राइट, फ्लैजेला आर हेयर लाइक आउटग्रोथ ऑफ द सेल मेम्ब्रेन. Cilia are the small structure which works, right, which work like an oar, right, causing the movement of either the cell or the surrounding fluid. Flagella are comparatively longer and responsible for the cell movement. The prokaryotic cells also possess flagella, but these are structurally different from the eukaryotic flagella. Electronic microscope study of the cilium and the flagellum shows that they are covered with the plasma membrane. They have a core, which is the aneuploidy, right? The number of microtubules running parallelly on the long axis, microtubules, and center mein we have the microtubules. Dr. Dark. Okay. Is it too much disturbing? Give me a minute. Yes, I think it's fine, bache. Just checked it. Yes. Very good. Chaliye. Now, here we have 9+2 arrangement रहता है. Central tubules are connected by the bridges and is also enclosed by the central sheet. वो देखा था हमने, right? There's an enclosed like this. And what we have next, which is connected to the one of the tubules of the each peripheral doublets by the radical spook, right? Thus, the 9 radical spook, we have total of 9. Peripheral doublets are also interconnected by the linkers. Both the cilia and the flagellum emerge from the centriole-like structure called as a basal body. Important line that we have over here in the NCERT. Moving further, we have the centrosome and the centriole. Centrosome is an organelle contained 2 cylindrical structure called centriole, surrounded by the precentrioles material. Both the centrioles in the centrosome lie perpendicular to each other and has the they are arranged in a kind of cartwheel. They are made up of 9 evenly peripheral tubulin proteins, right? And of course, they are called as a triplets. They have 9 of it and they are arranged in a 3. There are 3 and all together, and of course there are a total of 9. Adjacent triplets are also linked. The central part does not have, right, anything, and we call it as a hub. Clear? And it is connected with the tubules, again with the radical spook, made up of the proteins, and the centrioles form the basal body of the cilia and the flagella, spindle fibers, and give rise to the spindle apparatus during the cell division. Yahan pe hai, what we have the function of the centriole. What we have the function of the centriole, and this is the image that we have in our textbook. Everyone, all of you are clear? Yes, everyone, I hope that all of you are clear. Yaad rahega ye diagram? Yes, all of you, I hope that you will remember this. Clear? Quick thumbs up, everyone. I hope that all of you are enjoying the session. Please make sure you hit the like button for the video. Yes. Okay. The second last topic, everyone, which is what? Which is our nucleus. Nucleus के बारे में हम डिटेल में हमारी नेक्स्ट क्लास में पढ़ेंगे, which is cell division, cell cycle, right? तो अभी what we have very basic information, right, as compared to we'll be going into details in the next chapter, but here what we have, let's quickly see. Why does the nucleus have a membrane bound? Now this is a very question I'm sure you all have the answer for. Why does the nucleus have to be membrane bound? Can anyone tell me? Why we have the membrane around the nucleus? 1 minute, everyone. Yes, everyone. Why the membrane has to have a membrane? Yes. क्यों जरूरी है? Why does the nucleus have to be a membrane bound? Because of course, in the absence, if we don't have the membrane, it's an entirely different cell. So what's the difference between the prokaryotic and the eukaryotic cell? The major difference is that in prokaryotic cell, of course, they don't have a nuclear membrane. In the eukaryotic cells, we have a membrane, and that is the main reason of the differentiating between the two cells, right? Clear? So that's a very basic information that we have. What we have inside the nucleus is the genetic material, and this genetic material is super important. The nuclear membrane that we have protect the genetic material in the eukaryotes, right? Let's take a look, everyone, at the history of the nucleus. So Robert Brown in 1831, very important name, and the year I'm sure you would have seen long back previous year questions. May we have this question. Very confusing question, I would say, because we have Robert Hooke, Robert Brown, so people usually get confused, right? बच्चों को बहुत confusion हो जाता है यहाँ पे क्या सही आंसर है. और years, जितने भी years दिए हैं हमें वो बहुत important होंगे हमारे NEET examination के लिए. So Robert Brown, right, in the year 1831, observed the structure which is uniformly presented in the center, right, and we call it as the nucleus. तो ये हमें याद रहेगा. दूसरा, we have Fleming, right, in the year 1879. What he did, he actually used some basic stain. Basic stain use किया था, वो समझ में आया कि DNA जो अंदर genetic material क्या है, acidic है. Clear? And he observed that कुछ part stain हुआ है in the nucleus and he called that as the chromatin. इस तरीके से हमारे पास में नाम आया chromatin. Everyone, are you there with me? I hope that you can hear me. Yes? Nucleus का नाम किसने दिया? Robert Brown in the year 1831. And then we have chromatin. Chromatin का नाम किसने दिया हमें? Fleming ने in the year 1879. Stain करके देखा, वो जो stain part था वो दिख गया और इसलिए नाम दिया chromatin. Chroma means color, right? So यहाँ से connection हमारा समझ में आता है. फिर आगे अगर हम बढ़ेंगे तो हम बात करेंगे न्यूक्लियस को ब्रेन ऑफ द सेल भी कहा जाता है और इसके पास में क्या है, डीएनए है, जेनेटिक मटेरियल है. अपार्ट फ्रॉम दैट देयर आर वेरियस इंपॉर्टेंट स्ट्रक्चर दैट वी हैव इन द न्यूक्लियस. व्हाट वी हैव द न्यूक्लियोलस एट द सेंटर, वी हैव न्यूक्लियोप्लाज्म, we have nuclear envelope, the nuclear membrane, and we have the nuclear pores which are present for the exchange of the material. Apart from that, we call the nucleus as the brain of the cell. Important. And what we have the chromosomes also. So यहाँ पे अगर हम देखें, inside the nucleus we have the DNA, right? And all the processes, right? DNA replication, DNA की कॉपी होना, RNA बनाना, वो सब कहाँ पे हो रहा है? It is happening inside the nucleus, right? Everyone, are we clear? How many of you remember about the ribosomes? जल्दी से बताइए. Where in the cell there is a production factory of ribosomes? कहाँ पे है ribosomes की production factory? Yes, Dr. Duck. Yes, Ashutosh. Yes, everyone. जल्दी से. Where is the ribosomes factory present inside the nucleus? Jaldi. Very easy answer, right? We have talked about the ribosome. Yes. Very good, everyone. Where we have? We have it in the nucleolus, right? हमने पढ़ा है nucleolus. Nucleolus के अंदर क्या है? There is a ribosomes production factory. Important thing जो हम याद रखेंगे. Let's move ahead and let's talk about the nucleus. बहुत सारे सेल्स में क्या होगा, हमें एक सिंगल न्यूक्लियस भी रहेगा, ठीक है. बहुत सारे सेल्स में हमें दो न्यूक्लियस दिखेंगे और बहुत सारे सेल्स में हमें बहुत सारी न्यूक्लियस दिखेंगे, right? We'll see multinucleated. Now of course this also depends on the activity of the cell, right everyone? Clear? So this is, we have different, in there also we have one nucleus, two nucleus and multiple nucleus also. If you take a closer look at the nucleus, right, this is how it looks. And if you can clearly see this, right, they are attached to the ER. So if this is a nucleus, right, if this is a nucleus, right, so the ER is kind of attached like this. It is actually at the outer membrane, right? यहाँ पे दिख रहा है हमें. Clear? Nuclear membrane कैसा है? Double membrane है बीच में, right? And यहाँ पे जो outer membrane है वहाँ से we have the ER emerging. Yes? ये दूसरी साइड भी होगा. So this is how it looks under the microscope. So over here what we have the nuclear envelope जो हमें यहाँ पे दिख रही है. ये है हमारी nuclear envelope. Right, and we have the nuclear pore, and these pores plays a very important role in the exchange. जो यहाँ पे बन रहा है सामान वो बाहर कैसे जाएगा? With the help of the nuclear pore. यहाँ पे हमारा nucleoplasm act as a cytoplasm only, fluid है, इसके अंदर हमारे chromatin materials हैं. Then of course we have the chromatin जो nucleoplasm में ही है. Where have the nucleolus? Right everyone, what we have? We have the nucleolus and we have the ER. राइबोसोम्स यहां से निकल के फटाफट यहां पे चले गए, इजी पीजी, राइट? Now everyone, let's take a look at the structure, detailed structure of the nuclear envelope. Double membrane structure है, याद रहेगा हमें. So the nuclear membrane is double membrane. Nuclear envelope भी बोलते हैं. It's double membrane. It is associated with the ribosomes, right? Outer membrane पे what we have? We have just seen, right? On the outer membrane, what we have? We have the ER and of course we have ribosomes on it. Clear? Then it's a continuous with the ER, endoplasmic reticulum, and the inner membrane has two membrane, right? Inner membrane भी है. इसके अलावा इन दोनों membrane के बीच में जो gap है, जो distance है, right, वो कितना है? 10 to 50 nanometer. And this particular space is filled with the pre-nucleus. Fluid से भरा है, उसे क्या बोलते हैं? Pre-nucleus space. Clear everyone? क्या बोल रहे हैं हम उसे? Yes, pre-nuclear space. And इसका diameter यहाँ पे जो distance जो दिया है हमें, वो कितना है? 10 to 50 nanometer. Clear? इतना गैप है बीच में. Not the diameter, the gap. Yes everyone, सबको समझ में आ रहा है? See, यहाँ पे से देखने के बाद ना it could be that you might feel कि मैम बहुत difficult है, नहीं हो रहा, मैम कितना सारा पढ़ना है, कितना डिटेल में पढ़ना है. पर अगर हम एक एक स्टेप लेंगे, एक एक टॉपिक एक एक दिन में पढ़ेंगे, तो we will be able to clear this examination. Please don't worry. Please be with the teachers, come to the class regularly, and I'm sure, 100% sure everyone, you will be able to clear the exam. You just need to come to the class regularly and study. Yes? यही दो चीजें चाहिए ना हमें, और तो as a student, that's a best thing that we can do. Okay. Now that we are clear with this, let's move ahead and let's talk about K. Membrane क्या है? Continuous नहीं है. Nuclear pore भी बीच बीच में है. Nuclear pores are present. We have small gaps in the nuclear membrane, and ये जो pores हैं actually help करते हैं in the communication with the cytoplasm. Apart from that, we have the nuclear lamina, right? It's a meshwork. Can you see over here? Yes, it's a meshwork. Provide the mechanical support. Here we have. See, yes, sir? It provides the mechanical support. Now let's talk about the structure of the nucleoplasm. इसमें हमारा detailed structure जाएगा because we'll be discussing about the chromatin. Protoplasm enclosed by the nuclear membrane. ठीक है? Nucleoplasm क्या है? Of course, it's very similar to the cytoplasm, but ये nucleus के अंदर है, right? Also called as the chiroplasm or the chirolymph. Interesting names, everyone, right? अगर हम देखें protoplasm की definition क्या थी, जो हमें क्या याद है? The protoplasm is equal to the cytoplasm and the nucleoplasm. Basically, the living part of the cell, nucleus plus the cytoplasm. So we can say that protoplasm enclosed by the nuclear membrane, right? Also called as the chiroplasm, right? Jelly-like matrix hai, contain water, lipids, protein, and dissolved ions. Most importantly, has chromatin into it. Can we see this thread-like structures? These are nothing but the chromatin. And what is chromatin? Jaldi bataiye everyone, what is chromatin? Chromatin is nothing but, of course, right, when during the cell division, this is the chromatin that will be forming the chromosomes. So it's basically nothing but the composition. If you talk about the chromatin composition, it has DNA, it has histone proteins, non-histone proteins, and RNA. 4 important things are there, okay, and which is, of course, is suspended in the nucleoplasm. Furthermore, we have the nucleolus. We all know what is the function of the nucleolus, right? Clear everyone? Now, nucleolus ka, as I was mentioning earlier, what is the main function? It is a ribosomes factory, right? So it's a dense spherical structure in the nucleoplasm. Nucleoplasm ke andar, we have a dense structure which is called as a nucleolus. It is not bounded by a membrane. There is no membrane around it. Composed of DNA, RNA, and protein, and the site of ribosome assembly. यहाँ पे क्या हो रही है? Ribosomes की assembly हो रही है. Then of course, if you take a look at this, we have tiny thread-like structures, which is called as a nucleoprotein fiber. It contain what? Contain DNA. We have RNA, basic histone proteins and non-histone proteins. ये chromatin की बात कर रहे हैं. ये chromatin कहाँ पे है? Nucleoplasm में ही है ना? Right? So, yeah, we have these important things, right? And nucleolus की भी. Everyone, are we clear? Yes. Very good, Ash. Okay. So, now if we talk about the chromosome, we know that क्रोमोसोम्स आर मेड, क्रोमोसोम्स कब दिखते हैं हमें? will not say made क्योंकि तो होते ही हैं, but we'll be able to see the chromosomes only during the cell division. So the DNA when it gets coiled, right, it gets wrapped on the histones which are proteins, right, and they get more and more coiled further. Of course we call them as a chromatin fiber, and these chromatin fiber during the cell division forms the chromosome, right? In this also we have two different types of it, right? Chromatin fibers जो हैं, somewhere they are very loose and somewhere they are very dense. So the loose part, right, of the chromatin is called as the euchromatin. Euchromatin. Lightly stain है, अगर stain करेंगे तो lightly stain होगा, और heterochromatin है जो darkly stain होगा और densely packed है. So euchromatin and heterochromatin. अब अगर हम बात करें which of these is active, कौन सा जल्दी जल्दी ट्रांसक्रिप्शन में हेल्प कर सकता है, बाकी समय हेल्प कर सकता है? यूक्रोमाटिन, इट इज मोर एक्टिव. अब जो एकदम कॉइल्ड पार्ट है, राइट, दिस डेंसली कॉइल्ड पार्ट ऑफ कोर्स विल नॉट बी एबल टू अनवाइंड इजीली, तो ये थोड़ा शांत रहता है. दिस इज लेस एक्टिव. If you are in 11th class, it's okay, you will be learning more about this in class 12, and if you have watched our session, the molecular basis of inheritance, we have discussed it. Yes, we have discussed it. Villager, very interesting question, but we'll be able to tackle, of course, this. Now, genes की अगर हम बात करें, right, genes क्या हैं? They are the small fragment of the DNA active part. So, वो euchromatin and heterochromatin के बीच वाले ही के basis पे होंगे, right? Euchromatin क्या है? There is a light part of it, जिसे हम easily ट्रांसक्रिप्शन में यूज कर सकते हैं, जो एक्टिवली पार्टिसिपेट कर पाएगा बाकी प्रोसेसेस में, एज कम्पेयर टू द हेटेरोक्रोमैटिन जो कुछ भी पार्टिसिपेट नहीं करेगा. ठीक है. जीन्स क्या है? जीन्स कुछ नहीं, एक सीक्वेंस ही है, राइट. आरएनए जब बन रहा है हमारा, डीएनए के भी हमारे पास सीक्वेंस है, ATGC. आरएनए बन रहा है इन द प्रोसेस ऑफ ट्रांसक्रिप्शन, उसमें टी की जगह पे वी हैव द यू. अब जब आरएनए बन रहा है वो बाहर जाके प्रोटीन्स बनाएगा. क्लियर? तो जो हमारे जेनेटिक कोड्स हैं वो ऐसे ही बन रहे हैं. ठीक है. कोई स्पेसिफिक वी कैन नॉट कॉल इट, वी यूजुअली कॉल इट एज अ फ्रैगमेंट. ठीक है. एवरीवन आर वी क्लियर? यस, वेरी गुड, वेरी गुड. चलिए, नाउ एवरीवन, इट्स टाइम फॉर अस टू फिनिश द क्लास, राइट. वी नो दैट द क्रोमोसोम्स आर देयर, एंड ऑफ कोर्स वी हैव टू क्रोमोसोम्स. वन क्रोमोसोम वी आर गेटिंग, राइट, मतलब हर क्रोमोसोम एक पेयर में होता है. वन वी आर गेटिंग फ्रॉम अ मदर एंड वन फ्रॉम अ फादर, एंड ईच क्रोमोसोम हैज टू क्रोमैटिड्स, सिस्टर क्रोमैटिड्स, ये यहाँ पे. क्लियर? देन वी ऑल्सो कॉल देम एज अ होमोलोगस क्रोमोसोम्स. सिस्टर क्रोमैटिड्स बोलते हैं उनको. बीच में क्या है? वी हैव द सेंट्रोसोम. हियर वी हैव द सेंट्रोसोम. कैन वी सी ओवर हियर द सेंट्रोसोम, एंड साइड में व्हाट वी हैव, राइट, सेंट्रोसोम है. It will be holding the two chromatids, right? It divides the chromosome into the short and the long arm, P and the Q arm, right? And it is also called as a primary constrict. Constrict क्या होता है? थोड़ा सा bent होना, dent होना. तो यहाँ पे हमें दिख रहा है ना, क्रोमोसोम ऐसा है, तो यहाँ पे थोड़ा सा डेंट है, that is the primary constrict. Clear? उसके बाद में what we have? We have one more special thing on the chromosomes, which are called as the kinetochores, right? They are the proteins, disc. ये प्रोटीन के होते हैं, disc like structure है, and they are very on both the sides to the centromere, and they play a very important role in the cell division, right? They plays a very important role in the cell division. We will learn more about it in the next chapter. Each chromosome has the telomere. Telomere क्या है? The terminal end. यहाँ पे एकदम एंड पे, राइट, द टर्मिनल एंड्स ऑफ द क्रोमैटिड्स आर कॉल्ड एज द टेलोमर. एंड यहाँ पे वी हैव दिस वेरी स्पेशल स्ट्रक्चर, सेकेंडरी कंस्ट्रिक्ट रहता है. यहाँ पे से डॉट अगर हम बनाए तो सैटेलाइट बनी है. क्लियर? कैन वी सी ओवर हियर? दिस वी कॉल इट एज द सेकेंडरी कंस्ट्रिक्ट, राइट, एंड इट इज यूजुअली फाउंड एट द अदर ऑफ द प्राइमरी. मतलब प्राइमरी के अलावा वी हैव दिस, एंड इट्स यूजुअली देयर एट द एंड, एंड आर्म्स की एंड पे ही होती है. Clear? It's there only at end of the arm. And this chromosome have the satellite also. I made that right. छोटू सा satellite will be there. Okay. And here we have the satellite. Yes. What is satellite? Satellite क्या है? Part of the chromosome separated. Part of the chromosome ही है, पर किसने separate करा है उसे? Secondary constrict की वजह से वो थोड़ा सा दूर हो गई है. यहाँ पे ये रही satellite. राइट, सैटेलाइट है, इट्स अ पार्ट ऑफ क्रोमोसोम ओनली, पर थोड़ा सा अलग चली गई है, क्यों? क्योंकि सेकेंडरी कंस्ट्रक्ट की वजह से. Actually help in differentiating the different organism and help in the formation of the nucleolus after the cell division. So that's an important function of the. Everyone, are we clear? Yes, all of you. Are we clear with this? Yes, चलिए. Last part everyone, ये हम डिटेल में आराम से सेल डिविजन वाले चैप्टर में पढ़ेंगे. Then let's talk about the chromosome based upon their centromere. We have different names for the chromosomes, right? Sister chromatids, centromere अगर center में है तो of course we have it. We have a very special name, short arm P, long arm Q, and we have the telomeres, and we call it as different types, right? We have metacentric, there at the center. If थोड़ा सा P arm की तरफ ज्यादा जा रहा है तो उसे हम बोलेंगे सब metacentric. Acrocentric अगर एकदम ही end पे, मतलब end से थोड़ा सा पहले है, and telocentric which is there at the end. Okay, you can take a screenshot of this everyone. Yes, P arm and Q arm are equal in the metacentric. Then we have submetacentric, जहाँ पे थोड़ा सा Q arm ज्यादा बड़ी है और P arm से, P arm थोड़ी छोटी है, P arm देख रहे छोटी होती जा रही है, Q arm बड़ी है definitely, and यहाँ पे तो P arm है ही नहीं, छोटी सी भी नहीं है, there is no P arm. Okay, चलिए, with this everyone we are done. Yes, we are done with this everyone, so let's quickly read it through. I have one more class, so probably I will be rush a little bit, but everyone, next week we will have a quiz around it. Okay, we will have a quiz for all the important PYQs that we have from this particular chapter, so please be ready for it. Okay, चलिए, न्यूक्लियस है, हमें पता है बहुत इंपॉर्टेंट ऑर्गेनल है, रॉबर्ट ब्राउन ने 1831 में डिस्क्राइब करा था, फ्लेमिंग ने क्रोमैटिन नाम दिया था, इंटरफेज न्यूक्लियस में हम देख पाएंगे, राइट, द न्यूक्लियो प्रोटीन फाइबर्स कॉल्ड एज क्रोमैटिन, राइट, और कहाँ पे है? न्यूक्लियोली में, जिसे हम न्यूक्लियोलस भी बोलते हैं, राइट. वी हैव द इलेक्ट्रॉनिक माइक्रोस्कोप, बताता है हमें पैरेलल मेम्ब्रेन, इसके बीच में जो गैप्स हमने देखा था, राइट. पेरी न्यूक्लियर स्पेस है 10 to 50 नैनोमीटर की. Yes, then we have the outer membrane usually remain continuous to the ER. It has the ribosomes. Number of nuclear membrane, right? We have lot of minute pores all around it, which plays a very important role in the transportation, right, for the RNA and for the protein. What we have over here, nucleoplasm, contain the nucleolus in chromatin, right? Nucleolus and chromatin. The nuclei spherical present in the nucleoplasm. The content of the nucleolus is continuous with the rest of the nucleoplasm because it's a membrane. It does not have a membrane, right? It is a site of active ribosomal synthesis. It's there, or everyone, it's there in the textbook only. Larger and more numerous nuclei are present in the cells actively carrying the protein synthesis. Here we have the all information about the chromosomes. We'll not be discussing it, but I would encourage you to read it by yourself. Everyone, are we clear? All of you, are we clear with it? And of course, we have this. It's a homework question for all of you, everyone. Homework question for all of you. Right? Please do make sure you write the answer for this. And last topic, what we have the microbodies, which are single membrane bound organelles. They are single membrane bound organelle. They not a part of the endomembrane system. They are definitely not a part of the endomembrane system. Contain enzymes and found in both animals and plants. What is more important, we have three important types, and this is a PYQ question. Isko as it is yaad rakh lijiye. Sure, short PYQ question, right? We have peroxisomes, breakdown of the very long chain of fatty acids. We have spherosomes, synthesis and the storage of the lipids. Glyoxysomes convert fat into the carbohydrate. Super important, everyone. Take a screenshot of this, right? There are three main types of the microbodies that are present in both plants and animals. So here we have three types of microbodies that are present in both plants and the animals. Here we have question aata hai is pe. Clear? Yes. And again, everyone, here we have the. चलिए इसका आंसर बताइए. During the germination, अभी हमने देखा, right? Yes, during the germination, यस आशुतोष, बच्चे माइक्रो बॉडीज के बहुत छोटे छोटे स्ट्रक्चर हैं, राइट, सिंगल मेम्ब्रेन स्ट्रक्चर है, बोथ प्लांट्स एंड एनिमल में प्रेजेंट हैं, राइट, एंड दे हैव एंजाइम्स. क्या है इनके पास में? दे हैव द एंजाइम्स. चलिए, वेरी गुड. आंसर बी सही है इसका. अभी भी पढ़ा तो सबसे सही आंसर दिया. वेरी गुड. एवरीवन हियर द होमवर्क क्वेश्चन. सो वी हैव टोटल ऑफ 4 होमवर्क क्वेश्चन एवरीवन, राइट, 4 क्वेश्चंस इन टोटल. प्लीज डू राइट द आंसर इन द कमेंट सेक्शन बिलो. 1 2 3 4 करके यू राइट द आंसर्स, राइट. ठीक है, इस क्वेश्चन को हम 3 कर देते हैं, इस क्वेश्चन को 4 कर देते. फर्स्ट 1 2 3 4. क्लियर ऑल ऑफ यू? चलिए, यस, ओके मैम. बच्चे हिंदी में ही पढ़ाया था विकास, हिंदी इंग्लिश दोनों में पढ़ाया था. Yes everyone, now I have to leave. I have one more class. Thank you so much everyone. I hope that all of you have enjoyed the session and I hope that all of you have learnt about the nucleus, cilia, flagella, right? Next week हम इसका पक्के से टेस्ट लेंगे, तो please make sure you are preparing yourself for it. NCERT पढ़ के आना क्लास में, पिंकी प्रॉमिस है ना, राइट? पिंकी प्रॉमिस एवरीवन, प्लीज मेक श्योर यू रीड दी NCERT. फ्राइडे सैटरडे संडे आ रहा है, छुट्टी होगी आप लोगों की, नहीं है तो थोड़ा सा टाइम निकाल के फटाफट से NCERT पढ़ना. यस, चलिए. With this everyone, I'll end my session. Thank you so much everyone for being here with us. Lots of love and lots of care from our side and keep on learning with Aakash BYJU'S. Bye bye everyone. See you soon.